Assalamualaikum dear aimers. Hope so you all are doing well. I am Ariba Nadeem and the topic we are going to study is heme degradation. Very important topic, so listen to the topic very carefully. So firstly, we all know that RBCs have lifespan of 120 days. Why do we say that after 120 days RBCs are dead? Why do we say that? We say that because you all know that suppose this is RBC. Okay, I'm not good in drawing. This is RBC. RBCs have some proteins inside them, and these proteins help to maintain the flexibility of the cell membrane of RBCs. These proteins are, firstly, there is a protein which is termed as spectrin. Then there is a protein which is termed as, suppose these are encrins. And then there is a protein which is called as glycopharin, or you may call it as band 3 proteins, glycopharin. So these three proteins, what do? Is they make the cell membrane of RBCs flexible. That whenever the RBC passes through the capillaries, it can squeeze and it can easily cross the capillary. This is the function of these proteins. When RBC has completed its 120 days, then these proteins start becoming weak. Okay, and when these proteins start becoming weak, the RBC is no more flexible. The membrane of RBC is no more flexible. The membrane of RBC becomes rigid, and we say that now RBC is dead because this RBC is then taken up by the phagocytic system. So where does this RBC go after it becomes, after it has completed its 120 days? After the RBC completes its 120 days, it is taken up by mononuclear phagocytic system. Okay, mononuclear phagocyte system. What is mononuclear phagocyte system? It is basically same as that of reticuloendothelial system. If you know about that, reticular endothelial system is what? It is the part of immune system which consists of all the phagocytes of the cells, all the phagocytic cells present in the body. Okay, so mononuclear phagocytic system, the system consists of all the phagocytic cells, and all, as they all are mononucleate, so they are termed as mononuclear phagocyte system. For example, the Kupffer cells present in liver, that is included. The macrophages present in spleen, that includes in reticuloendothelial system or macrophage, mononuclear phagocytic system. Similarly, there are macrophages present in skin, there are macrophages present in your brain, microglia. So all these macrophages, they are collectively called as, all these phagocytic cells, they are collectively called as mononuclear phagocytic system. Okay, so after the RBC completes its 120 days, it is taken up by this mononuclear phagocyte system. And what is mononuclear phagocyte system? It is a part of immune system which consists of phagocytic cells. Okay, which phagocytic cells take these RBCs? Out of many phagocytic cells which are present in skin, liver, brain, spleen, all that, the RBCs are taken up by the phagocytic cells of liver and spleen, and mainly spleen. That is why the spleen is called as the graveyard of RBCs, because RBCs are degraded in spleen. Okay, so the macrophages present in liver and spleen, they are responsible for taking these RBCs who have completed their 120 days. Now, this is RBC. RBC is containing hemoglobin. This hemoglobin consists of two parts, a heme portion and a globin portion. This heme portion is basically a non-protein part, and this globin portion, the globular, always referred to, the proteins in a globular shape. Okay, so globin refers to a protein part which includes 2 alpha and 2 beta chains. Now we will study in detail the heme part, what happens to the heme part of RBC when RBC has completed 120 days. But just a quick overview that this globin part, which is converted into amino acids, okay, this globin part is basically converted into amino acids with the help of peptidases. So peptidases are the enzymes which convert globin part into the amino acids, and these amino acids are then recycled back. Okay, they are again used in the synthesis of RBCs because they are again used to make globin, they are again used to make hemoglobin. Okay, so they are recycled. What happens to this heme portion? Firstly, be clear about heme portion. It's a non-protein part. It's a prosthetic group which is attached to the hemoglobin. Hemoglobin is a protein part and heme is a non-protein part. So when a non-protein part is covalently or tightly bound to the protein part, then it is referred to as prosthetic group. So heme is basically a prosthetic group of hemoglobin, and 85% of the heme which is degraded is basically degraded by this hemoglobin present in RBCs. Otherwise, heme is also present in other compounds like cytochromes, like catalase, like myoglobin. Heme is also present in that compounds, but 85% of the heme is degraded by the heme present in the hemoglobin of the RBCs, old and worn out RBCs, you may call it as this. Okay, so now we will see how this heme degradation will occur and what products will be formed by the degradation of this heme. Okay students, till now we have studied that we have RBC which has completed 120 days. It contains hemoglobin. This hemoglobin is degraded into two portions, globin and heme. The globin is converted into amino acids by peptidases, and these amino acids are then recycled. What happens to the heme portion? That this RBC was taken up by the macrophage of liver or spleen. Okay, now the first step in the heme degradation is that this heme contains porphyrin ring. So first step is the unwinding of this porphyrin ring. This porphyrin ring is converted into a linear structure. I'll name that linear structure later. So what happens that firstly, the most important component in heme was iron plus 2. Okay, so firstly iron plus 2 is removed from the heme. Okay, and where does that iron plus 2 goes? That iron plus 2 is stored in the form of ferritin in the body. The ferritin is the storage form of iron. So this iron plus 2 is stored in the form of ferritin in the body. Then what comes out next is carbon monoxide. There are two things which are removed from the heme, and there are three things which are added into the heme in the first step of the heme degradation. The two things which are removed from the heme, the first is the most important component of heme, which is iron plus 2, which is stored in the form of ferritin, and the second component is carbon monoxide. It is also very important. I'll tell you its importance later. And what are three components which are added into the heme? This heme is basically converted into a linear substance. This porphyrin ring opens up and it forms a linear substance which is called as biliverdin. Biliverdin. For the formation of this biliverdin, what do you need as? For the formation of biliverdin, you need 3 oxygen molecules and you need 7 electrons. Okay, so what we are going to add first is, this is oxygen. Okay, so I'm going to add oxygen over here. As I need to add oxygen, so I need some kind of enzyme. So there is an enzyme which is referred as heme oxygenase. What is that enzyme? Heme oxygenase, which basically incorporates oxygen into the heme to make biliverdin. And the third thing we need to add is 7 electrons. We all know that if we need to add electrons or hydrogens, the only source we have is NADPH. Okay, so this NADPH is converted into NADP plus. As it has got a plus sign, it means it has donated the electrons. Okay, so those 7 electrons, those oxygen atoms are added into the heme with the help of heme oxygenase. Iron plus 2 and carbon monoxide is removed from the heme, and there is the formation of biliverdin. Do remember that this heme is red in color, while this biliverdin is green in color. As it has added oxygen into it, as it has added electrons into it, so this is the oxidation step. Okay, so biliverdin is basically green in color. It has no iron plus 2, while heme has iron plus 2, and it is the oxidized product of heme. Okay, oxidized product of heme. So this is the first step. You have to remember, you need to exclude 2 things from the heme and you need to add 3 things into the heme. And then you have to remember that this heme is red in color, while this biliverdin is green in color. It has no iron plus 2 and it has basically formed by the oxidation of the heme. Now, what is the function of this carbon monoxide formed over here? What happens that whenever RBC undergoes, the hemoglobin present in RBC undergoes degradation, then there are chances of hypoxia, because RBC number can decrease and the hypoxia can occur. Now, what does hypoxia do? Hypoxia always triggers inflammatory mediators. Hypoxia triggers, I'm going to write over here, it triggers inflammatory mediators. Means the chemicals or the substances which cause inflammation. It triggers inflammatory mediators and ultimately it causes inflammation. It can lead to inflammation. So what nature has done over here, that it has made a molecule carbon monoxide. What does this carbon monoxide do? This carbon monoxide is basically a signaling molecule. You will find the line from wherever the book you are reading. You will always find the carbon monoxide is a signaling molecule. What signal does carbon monoxide transmits is that it signals the inflammatory mediators and orders them to inhibit. So carbon monoxide basically inhibits inflammatory mediators. Inhibits inflammatory, I'm just writing IM, which means inflammatory mediators. So this carbon monoxide basically inhibits these inflammatory mediators, and the second thing it does is that it increases the production of anti-inflammatory mediators. Increases production of anti-inflammatory chemicals. And what are the anti-inflammatory chemicals? Like cytokines like interleukin 10. This is anti-inflammatory, which prevents inflammation. Okay. So what does this carbon monoxide do? That this carbon monoxide inhibits the inhibits inflammatory mediators and increases the production of anti-inflammatory chemicals like interleukin 2. These both things prevent inflammation. So it acts as an anti-inflammatory substance. And when there is no inflammation, there would be no apoptosis, because in apoptosis there is inflammation. It is anti-apoptosis, and thus it protects the cell, so the cell will be protective.